Okay, so welcome back to this next video in which we are discussing anxiety and anxiolytic drugs. Okay, so we've now discussed the antidepressants, and it is fair to say that the antidepressants are the first line treatments for anxiety. Okay, and that means that they are the first choice that a doctor would use in the treatment of anxiety. They take 3 weeks before they actually start to work, however. We're now going to discuss the second line treatment, okay, which will be used if the antidepressants don't work, okay, and the other case where you would use this second line treatment is if you need very, very acute remission from an anxiety attack, so from a panic attack, for instance, okay, because these second line drugs, these work within 30 minutes rather than 3 weeks. Okay, so we're going to talk about the benzodiazepines, so I'll just get a new piece of paper. Alright, so we're going to start off by discussing the benzodiazepine structure, okay, so the structure that's within all of the benzodiazepines, okay, because it's actually a chemical definition, not a biological definition based on their actions, okay, and then what we'll do is talk about how these actually work by promoting GABA-ergic transmission. Okay, so benzodiazepines then. Right, so to discuss the structure of benzodiazepines, we need to firstly discuss what a diazepine ring is. Okay, so a diazepine ring is a 7-membered ring which contains 2 nitrogen atoms and 5 carbon atoms. So I'm going to put the first nitrogen up here, and it will have a hydrogen coming off. And again, I'm going to draw a skeletal structure here. Okay, so now let me draw the 7-membered ring. So, here is a carbon, and here potentially is a carbon, okay, because I'm not going to put in the second nitrogen at the moment, so I'll just tell you how I'm going to do this. I'm not going to initially put in the second nitrogen, because the position of the second nitrogen can vary depending on what kind of diazepine ring you want to make. So at first I'm just going to put them all as carbons, but that's not a diazepine ring yet. Okay, so here's a carbon, here's a carbon, here's a carbon, here's a carbon. Okay, then we go on like so. And that gives us a 7-membered ring. 1, 2, 3, 4, 5, 6, 7. Okay, and you're going to have a double bond here, a double bond here, and a double bond here. Now, that is not yet a diazepine ring, because at the moment we only have 1 nitrogen. One of these carbons needs to be replaced with a nitrogen. Now, if you choose to replace this carbon here with a nitrogen, the diazepine ring that you then get will be known as a 1,2 diazepine ring, okay, because the 2 nitrogens are in position number 1, which is this one, and then position number 2, okay. If instead you choose this carbon here to replace and put a nitrogen there in that position, then it will be a 1,3 diazepine ring, because the 2 nitrogens will be in position 1 and position 3. So this is position 1, then you'll count 2 and 3. Okay, if on the other hand you pick this position here, okay, to substitute in your nitrogen, then that will be what's known as a 1,4 diazepine ring. Now, you don't at any point get rid of the double bonds in this structure when you replace the carbons with nitrogens. Remember, each of these carbons here will have a hydrogen coming off it. So when you replace that carbon with a nitrogen, all you do is you don't put a hydrogen onto the nitrogen, because remember, nitrogen only needs 3 bonds, whereas carbon needs 4 bonds. Okay, so all you do is you remove the carbon with the hydrogen coming off and replace it with a nitrogen atom. Now, you might say, well, what about replacing these ones? Well, replacing this one is exactly equivalent of replacing this one is exactly the equivalent of replacing this one, and replacing this one is exactly the equivalent of replacing this. So they'd still be called the same things, basically. Okay, so 1,2 diazepines, 1,3 diazepines, and 1,4 diazepines. In benzodiazepines, you are going to use the 1,4 diazepine ring. Okay, so let's now redraw out a 1,4 diazepine ring, and this time we'll draw it out properly where we have both nitrogens in the ring. Okay, so here's our first nitrogen. Okay, these two now will both be carbons. Okay, then we'll have another carbon here, and then this is going to be a nitrogen here. Okay, now all we need to do is put in the double bonds. So we'll have a double bond there, a double bond there, and a double bond there. And that now is our 1,4-diazepine ring. Now at the moment that's just a 1,4-diazepine ring. How do we turn this into a benzodiazepine structure? Well, the clue is in the name. What we are going to do is we're going to fuse a benzene ring onto this 1,4-diazepine ring, and that will create us a benzodiazepine. Okay, so we're going to put a benzene ring off this position here. Okay, so we're going to attach an additional 4 carbons on here, which will have alternating double and single bonds like so, and that will now create us a benzodiazepine structure. So in all of the benzodiazepine drugs, you'll have one of these, basically, and that's the characteristic feature that they all have. Okay, right, so let me now give you some examples of benzodiazepine drugs. So the first one that was discovered by accident years and years ago by Hoffmann and Roche is known as chlordiazepoxide. Okay, the most famous one of them all is diazepam. Okay, this is a very, very famous drug. Again, like fluoxetine, you probably won't recognize it until I give it its trade name. Its trade name was Valium. Okay, so that's the infamous trade name of this drug. Okay, so Valium is a very famous drug. Okay, then other benzodiazepines are clonazepam and lorazepam as well. So we try and end their names with zepam, although obviously it doesn't work all the time. Okay, clonazepam. Okay, and then also you have lorazepam. Right, so those are some examples of benzodiazepine drugs, and again, like with the antidepressants, there are loads more that I could give as examples, but we've got the most important one there, certainly. Okay, now what we want to see is how do these drugs actually work to treat anxiety, and what's incredible is that they work within 30 minutes. Now, we're going to see that really they work by lowering the function of every single bit of the brain, okay? And then their anxiolytic effects are probably just because they then lower the activity in the amygdala. But it's not that they selectively lower the activity in the amygdala, really they just lower the activity everywhere in the brain, okay? And that, hence, by brute force, they do lower the activity in the amygdala, and that's how they probably help with the anxiety. Okay, but because of the fact that they lower activity pretty much everywhere in the brain, they also have significant sedative effects. Okay, so they make you sleepy. Okay, so they are actually also used as hypnotic drugs, which means drugs that make you go to sleep. Okay, in the treatment of insomnia. Okay, so they're a hypnotic. Right. So how then do they lower the activity of all portions of the brain? Well, to understand this, we need to discuss GABAergic transmission in more detail, okay, because benzodiazepines are going to act on what are known as the GABA-A receptors, okay, but to understand anything about that, we need to understand GABAergic neurotransmission, and to understand GABAergic neurotransmission, we need to have a very decent understanding of classical neurotransmission. So, let me just again remind you of classical neurotransmission. So let me draw out a neuron here. Okay, so these are the dendrites of the neuron here. Okay, and all over the dendrites, although I'm not showing many of them, I'll show one of them here. You have these little processes coming off the dendrites of neurons, one of which is shown here, which are known as dendritic spines, and it is these processes which are involved in synapses. Okay, so these are the dendrites of the neuron here. Here is the nucleus of our neuron here. Okay, and then we'll have the axon here. Okay, going along and then ending with an axon terminal here. And the axon might be myelinated, so we'll put some myelin sheaves on here as well, just to make it look a little bit more like a neuron. Right, there we go. So, let's label up the portions here then. So this is the axon portion here. here is the axon terminal here, which is where this neuron itself will release neurotransmitter from. Okay, and then here we have the dendrites. These are very important. These are what collect information from other neurons and decide whether this neuron is going to fire an action potential. And the dendrites will be absolutely covered in little processes like this. I haven't shown them covered in little processes like this because it would have taken me absolutely ages to draw. Okay, but really they should be covered in tiny little processes like this sticking off from them, which are known as dendritic spines. Okay, and it is these structures which are involved in synapses. So basically, the axon terminal from another neuron is going to come and synapse on the dendritic spine, or a dendritic spine, of this neuron here. Okay, so I'll label the full thing up as a neuron. Right, so let's add a little bit of colour. So we'll have, well actually I don't want to put colour in because at the moment it's nice and clean and I can put lots of other things on without, and they'll show up. Okay, right, so the key thing to understand is that there are two major neurotransmitters in the brain. One is an excitatory neurotransmitter, okay, and that is glutamate, okay. If an axon terminal releases glutamate onto a dendritic spine, it will act on receptors for glutamate, which will be ligand-gated ion channels, and when they open, they will allow in positively charged ions from the extracellular fluid into the inside of the neuron. So generally there'll be sodium ions. Okay, they're going to allow sodium ions to come into the inside of the neuron. Okay, and all over the dendrites of the neuron, you're going to be getting these sodium ions moving in from synapses which are releasing glutamate. Okay, and these sodium ions are going to help activate this neuron to fire an action potential. Now, where does the action potential begin in most neurons? Well, the action potential does not usually begin in the dendrites. Instead, there is a special place where the action potential usually begins, and this is known as the axon hillock. Okay, so it's this beginning of the axon over here, okay, known as the axon hillock. Okay, and what's going to happen is the sodium ions that are coming in due to glutamate all over the dendrites of this neuron are going to diffuse over to the axon hillock, or at least a little bit of them is going to diffuse over to the axon hillock, and is going to promote the formation of an action potential at the axon hillock. So positively charged ions are going to promote the formation of an action potential at the axon hillock. So you're going to be getting positively charged ions coming in from all over the place from these synapses where glutamate has been released onto this neuron. Okay, and those are going to be promoting action potentials. However, at other synapses, so let's say here's another dendritic spine, and here we've got another presynaptic neuron, you might be releasing the major inhibitory neurotransmitter of the brain. And the major inhibitory neurotransmitter of the brain is GABA. And what this stands for is gamma, okay, and I've put it in Greek letter, but if I write it out in full, it'll be gamma, so that's the G, okay, and then it's amino, that's the A, and then the B is butyric, okay, and then the final A is acid. So this is gamma-amino-butyric acid, or GABA. Okay, and let me just show you the structure of this, because it's a very, very simple molecule. Okay, so firstly, butyric acid. Butyric acid is just the old name for a molecule that would now be called butanoic acid. Okay, so butyric acid or butanoic acid, they're the same thing. Okay, so that just means a 4-carbon carboxylic acid. So let me show this. Here's the carboxylic acid group, and then we just need 4 carbons, so let's put 3 more here. Okay, and then you're going to have hydrogens coming off these. Okay, like so. So it's just a 4-carbon, fully saturated carboxylic acid. Now, if I was just showing pure butanoic acid, I would put another hydrogen here. But I want to show GABA, not just butyric acid. Okay, so now gamma amino, what does this mean? This means that we're going to have an amino group coming off the gamma carbon. So what is the gamma carbon? Well, the old way of naming up the carbons in a carboxylic acid is to call the first carbon off the carboxylic acid group the alpha carbon. So this one would be the alpha carbon here. Okay, so this one here in red, this is the alpha carbon. Then the second one here would be the beta carbon, and then this one would be the gamma carbon. Okay, so when people say gamma amino, this means that you've got an amino group coming off the gamma carbon. So let me now just put an amino group there. Okay, so here's an amino group. So this is the structure of GABA then, or gamma amino butyric acid. Okay, so that's why I said it was such a simple structure. Right, so this is the major inhibitory neurotransmitter on the brain. Okay, so if this axon terminal here releases GABA onto the dendritic spine, it will act on receptors on the surface of the dendritic spine, known as GABA-A receptors, which we're going to discuss in more detail in a moment. Okay, so GABA-A receptors. Okay, or for short, GABA-ARs, R is for receptors, okay, and these receptors will open, they are again ligand-gated ion channels, and instead of allowing positively charged ions into the cell, they're going to allow negatively charged ions into the cell. Okay, so here come in negatively charged ions in the form of chloride anions. Okay, these are going to be diffusing over to the axon hillock as well, and they will be making it less likely that you will be firing an action potential. Okay, so all the time, neurons are taking all of this information from all over their dendrites. They're getting activatory signals coming in, and they're getting inhibitory signals coming in, and the two are cancelling each other out at the axon hillock. However, how do you then ever fire an action potential? Well, if the balance is tipped, if you suddenly get a huge amount of stimulatory input onto this neuron, then the stimulatory currents, the sodium ions, will beat the inhibitory currents, and then you'll fire an action potential. So it's a tip in the balance, basically. Okay, so basically what benzodiazepines are going to do is they're going to up the amount of inhibitory chloride anions that are coming into every single cell in the brain, and therefore they're going to tip the balance in favour of the inhibition, and they're going to make it much more difficult for neurons to actually be activated. So every single neuron in the brain is going to have its activity reduced, basically. It's going to become much more difficult to activate. So basically you calm down the entire brain, and you might say this sounds awfully dangerous, and indeed it is. If you overdose on these drugs, they will kill you by stopping your brain from actually functioning at all. Okay, so let's now discuss the GABA-A receptors in more detail, and then we can see exactly how the benzodiazepines are going to increase the amount of inhibitory chloride anions that you are getting coming into neurons. Okay, so let's study the GABA-A receptors then. Okay, right. So there is not just one GABA-A receptor, is the first thing that I need to say. It is an entire family of receptors. There are absolutely loads of the things, which is what I hope you're going to understand by the end of this discussion. Okay, now there are another family of GABA receptors as well. There are the GABA-B receptors. Those are much more rare. The GABA-A receptors are ligand-gated ion channels that open and allow chloride anions into the cell. The GABA-B receptors are G-protein coupled receptors and mediate mainly presynaptic inhibition. Okay, right. So, what then is the structure of the GABA-A receptors? So, once again, I will put two lines parallel to represent the inner and outer layer of phospholipids of the cell membrane. Okay, and now I'm going to draw a GABA-A receptor sitting in the cell membrane. Okay, and whenever you're drawing a ligand-gated ion channel, it's a good idea to start by drawing a cylinder of some sort. Okay, so here is a ligand-gated ion channel, here's the pore down the middle, and it's sitting in the cell membrane like so. This we'll say is the extracellular fluid side here, okay, and this is the cytoplasmic side. So here we are looking at the membrane of the dendritic spine. So to go back to this picture, we are looking at this postsynaptic membrane here of the dendritic spine, which is going to have the GABA being released onto it by that presynaptic neuron. Okay, right. Now, GABA-A receptors are not, repeat, are not just made up of a single protein. Okay, instead they're made up of 5 proteins joined together in a complex. Okay, so let me show this. Excuse me. Okay, so let me show this. So you're going to have 5 separate subunits making up the GABA-A receptor. So I'll divide it into 5/5 like so. Okay? So each one of these is a separate protein, and ideally they would have been the same size. You know, this one looks much bigger than this one. That's just the picture, basically. There are 5 equivalently sized proteins that are making up this GABA-A receptor. So 5 of them have come stuck together to make the full receptor. Okay, so it's what we would know as a pentamer. Okay, pentamer just means 5-membered structure. Okay, right. So now let's say we're going to pull out one of the individual subunits of the GABA-A receptor. Okay, so we're going to pull out one of these individual proteins which makes up 1/5 of the GABA-A receptor, and we're going to study the structure of that individual subunit. Okay, right, so let's have a look at the structure that you find. So here once again is the cell membrane. Okay, and we'll say that the top is the extracellular fluid again, and the bottom is then the cytoplasmic side. Right, so now we're looking at an individual polypeptide. So here is its amino terminus. Okay, so we're looking at a GABA-A receptor subunit. So the whole thing is called the GABA-A receptor. The individual proteins that make up the GABA-A receptor are known as the GABA-A receptor subunits. Okay, so for short, GABA-A receptor is abbreviated to GABA-AR, and this is a GABA-A receptor subunit. And we are now pulling a GABA-A receptor subunit out and having a look at the structure of a GABA-A receptor subunit. So, once again, this line will represent a polymer of amino acids. Now, on the extracellular side, you have a very key structure, okay, as far as the classification of these receptors is concerned. Okay, you have a little loop in the polypeptide structure that is held together by a disulfide bond between 2 cysteine residues, and is hence called a cis loop. Now, I want to make sure that everyone is fully with me with what this actually is. So I'm going to draw out a bigger picture. So basically, a cis loop is just a loop in the polypeptide structure, just like this, a hairpin loop. Okay, so here I'm drawing it out bigger now. So here, this line represents the polymer of amino acids. So it represents amino acid after amino acid after amino acid after amino acid, and now I'm suddenly going to draw one of the amino acids out in its full structure. Okay, so it's going to end up looking much bigger, but it shouldn't really. Okay, so this is going to be a cysteine amino acid, and it's going to be involved in holding the full thing together. Okay, so here's the amino group, here's the alpha carbon, here's the carboxylic acid group, and of course this is an amino acid residue, so I've drawn it as though it's bound within the polypeptide. Okay, and now the R group of a cysteine residue consists of a methylene group, and then with a thiol group coming off it. Now a thiol group really consists of a sulfur atom with a hydrogen atom coming off it. Okay, so this is what's known as a thiol group. Okay, and if you're unfamiliar with the chemical properties of thiol groups, the thing that I can say that will help you to understand them is that they have very similar chemical properties to alcohol groups, which hopefully you are familiar with. Okay, and the reason they have so similar chemical properties to alcohol groups is that sulfur is actually in the same group of the periodic table as oxygen. Okay, so sulfur has similar chemical properties to oxygen. If that sulfur was an oxygen, then that group would be an alcohol group. Okay, we're saying that the sulfur has similar chemical properties anyway to the oxygen, so this surely must therefore have very similar chemical properties to the alcohol groups. Okay, now I'm not actually going to show the hydrogen coming off the sulfur atom because I'm being fortuitous. This cysteine residue is actually going to be involved in binding to another cysteine residue on the parallel strand of the loop. Okay, and it's the thiol groups that are going to bind to one another, and in that binding they're going to lose their hydrogen, so that's why I'm not showing the hydrogen coming off this sulfur atom here. Okay, now I'm going back to showing the polypeptide just as a line, okay, and it's going to loop around like so, and then on the other side we're then going to have another cysteine residue here. So we'll have the amino group shown here, the alpha carbon here with a hydrogen coming off it, and then the carboxylic acid group here, and then you'll continue on with the polypeptide as so, and then the R group once again is going to be a methylene group, and then you'll have a thiol group but the thiol group once again loses its hydrogen so that these two sulfur atoms can bind together like so, and this special bond that you've got here is known as a disulfide bond, or it's also sometimes called a disulfide bridge. Okay, and it's a very, very strong bond. Okay, it's a nice covalent bond. Right, so this is what holds the whole cysteine loop together, or the whole cis loop together, and that's why this is known as a cis loop, because cis is short for cysteine, and it is the cysteine residues which are holding the loop together. So that's what that structure there is. So that's a nice detailed explanation of what that actually is. Okay, right, then what you have is you have a membrane spanning alpha helix, followed by a second membrane spanning alpha helix, followed by a third membrane spanning alpha helix with a very large intracellular loop between the third and then the fourth membrane spanning alpha helix. And then finally you finish with carboxylic acid tail here. Okay, so let's highlight up some of the key features that I've just told you about. Okay, so you have these 4 membrane spanning alpha helices, now coloured in green there. Okay, and these are all given names. The first one is called M1. Okay, the second one is called M2. The third one is called M3. And the fourth one is then called M4. Okay, right. Then, the large loop that you have between M3 and M4, that's quite important. That's the site of many post-translational modifications that regulate the function of the GABA-A receptor. Okay, this is what's known as, either you can call it the M3-M4 loop. Okay, that's very intuitive, or you can call it the intracellular loop. And for short, intracellular loop can be abbreviated to ICL. I for intra, C for cellular, and then L for loop. Okay, right, so either of those names you can use for this loop between M3 and M4. Okay, and then the final other key structure which we've just talked through is this very important cis loop on the extracellular side. So, GABA-A receptors, they all have subunits that have this same basic structure here. Now, there is not just one GABA-A receptor subunit, unfortunately. The really simple thing would be if there was one protein called the GABA-A receptor subunit. It looked like this, and you just had to take 5 of these GABA-A receptor subunits, stick them together, and you've got your GABA-A receptor. It's not that simple, unfortunately. Instead, there are actually 19 different GABA-A receptor subunits. Okay? Now, all of these 19 different GABA-A receptor subunits, they will all have different sequences of amino acids. Okay, so if you look at their primary polypeptide sequence, it will be different. Okay, however, they are similar enough that they all fold up into this basic structure. So they all have these same core features that I've just described to you. Okay, even though they are all fundamentally different. So they all look like this. Okay, so that is at least some good news, that they all have the same basic structure. Okay, right. Now what I'm going to tell you about is the naming of these 19 different GABA-A receptor subunits. So firstly, there are 6 different alpha GABA-A receptor subunits. So 6 of the GABA-A receptor subunits are known as the GABA-A receptor subunit alpha 1, the GABA-A receptor subunit alpha 2, the GABA-A receptor subunit alpha 3, and so on, all the way up to the GABA-A receptor subunit alpha 6. Okay, so that's 6 of the 19 already dealt with. Okay, then we have some beta GABA-A receptor subunits, and you have 3 of these. So you have the GABA-A receptor subunit beta 1, the GABA-A receptor subunit beta 2, and also the GABA-A receptor subunit beta 3. Okay, moving on, you also have gamma subunits. So you have the GABA-A receptor subunit gamma 1, the GABA-A receptor subunit gamma 2, and the GABA-A receptor subunit gamma 3. Okay, now you also have the GABA-A receptor rows. So you have GABA-A receptor row 1, GABA-A receptor row 2, and GABA-A receptor row 3. Now how many are we up to now? So you've got 6 here, you've got 9 here, that takes us up to 15, so we've got 4 left. These are the ones which aren't put into families in this way. We've got an alpha family, a beta family, a gamma family, and a rho family. The final ones are on their own. So there is the GABA-A receptor subunit delta, there is the GABA-A receptor subunit epsilon, and then there is also the GABA-A receptor theta and pi. Okay, so that is the naming of the 19 different GABA-A receptor subunits, and I will reiterate the point that they will all have different sequences of amino acids, so they are all different polypeptides, but they all have this same basic structure shown in this picture, and they can all form a fifth of a GABA-A receptor. Okay, so now it becomes a total nightmare. Okay, you have the challenge of building a GABA-A receptor. You have 5 separate sockets. You can pick GABA-A receptor subunits from this pile. That looks as though there is an absolutely nightmarishly big number of GABA-A receptors. Okay, and there is in the brain. Certainly not all of the different types that you can build are expressed within the brain. Okay, but a lot are expressed within the brain. However, thankfully, the brain tends to stick to a certain protocol when it's making GABA-A receptors. Okay, so there's generally a certain way that the brain likes to build GABA-A receptors, and it's this way that I'm now going to describe to you. I cannot go through every single GABA-A receptor that exists within the brain, there's too many. Okay, but I'm going to tell you the basic rules that usually brain cells use when they're going to build a GABA-A receptor. So the general type of GABA-A receptors only actually use GABA-A receptor subunits of the alpha, beta, and gamma families. Okay, they don't use these more exotic ones down here. Okay, so for that reason, these GABA-A receptors built out of alpha, beta, and gamma family GABA-A receptor subunits are known as alpha, beta, gamma receptors. Okay, right, so let me show you how you build these things then. So we'll now look at the GABA-A receptor as though we're looking down from above. Okay, so let me just put you in perspective. We are looking down from the extracellular fluid down at the GABA-A receptor pentamer from up high. Okay, so here is the pore. Okay, and we've now got these 5 separate GABA-A receptor subunits here. Okay, there's 3, 4, and 5. Okay, right. So in the main form of GABA-A receptor pentamers, what you do, this is the rule for how you build this alpha, beta, gamma type of GABA-A receptor. Firstly, you pick one of the 6 alpha GABA-A receptor subunits. So you pick one of these 6 alphas to use. Okay, you only pick one, and then you make 2 copies of that protein, and you put these 2 identical copies here and here. Okay, and I want to stress that these 2 alpha subunits that you are putting here, they are the identical type. You do not put one alpha 1 in one of these sockets and then an alpha 2 in another socket, or at least the brain doesn't. This is the general rules that it uses. It picks one of the alpha subunits that it likes the look of, and it uses that in both of these sockets. It does the same thing for the beta family. It picks one of those 3 beta subunits, and it uses that beta subunit in this socket and this socket here. Okay, let's colour these in green here. Okay, and then finally, the final socket that's left to fill, that will be one of the gamma subunits. So you'll pick one gamma subunit and you'll put it in here. Okay, so those are the rules by which you create one of these alpha, beta, gamma GABA-A receptors. Okay, and it gets even better. The even better news is that there is a specific alpha, beta, gamma GABA-A receptor pentamer that the brain particularly likes. The most important GABA-A receptor that's found most of places, okay, is one of these alpha, beta, gamma GABA-A receptors. And the alpha subunit that you use in this alpha, beta, gamma GABA-A receptor is the alpha 1 subunit. The beta subunit you use is beta 2, and the gamma subunit you use is gamma 2. Okay, so you pick this one as your gamma subunit. Okay, you pick this one as your beta subunit, and you pick this one as your alpha subunit. So the alpha 1, beta 2, gamma 2 GABA-A receptor is the main GABA-A receptor that you have within the brain. Okay, so these are the main types of GABA-A receptors that you have, and of them, the most common one is to have alpha-1 here, beta-2 here, and gamma-2 here. Okay, right, so if there is one GABA-A receptor that you are going to remember, this is the one to remember, basically. Most GABA-A receptors are this type of GABA-A receptor. Okay, right, now, going back to the more general alpha-beta-gamma GABA-A receptor, because all of those alpha-beta-gamma GABA-A receptors, they all have the same basic properties. And what I mean with regards to that is that they all have the same the GABA binding sites are the same. Okay, so for all alpha-beta-gamma GABA-A receptors, you have two GABA binding sites, and these GABA binding sites are located in between the alpha and the beta subunits of the GABA-A receptor. Okay, so this is a GABA binding site, and also you've got another GABA binding site up there, and you'll notice that they're positioned in between the alpha and the beta subunits of the GABA-A receptors. Okay, right, so that's the case not only for this alpha-1, beta-2, gamma-2 GABA-A receptor, but it's also the case for all alpha-beta-gamma GABA-A receptors. Okay, so overall then, to put this in perspective, you have these GABA-A receptors, which are mainly of the alpha-beta-gamma type, on your postsynaptic membranes of your dendritic spines. Okay, so these things are in the membranes of the dendritic spines. When GABA is released from the presynaptic neuron here, it will diffuse across the synaptic cleft, and it will now bind to these GABA-A receptors on the surface of the dendritic spine. Okay, specifically, two GABA molecules will bind to the GABA-A receptors, which are mainly going to be of this alpha-beta-gamma sort. Okay, so one GABA here and one GABA here, that will cause the receptor to open, so the pore will get much wider, and now chloride anions will move from the extracellular fluid into the cell, and that's the inhibitory chloride current, basically, that's going into the cell. Okay, right, now let's talk about benzodiazepines and what they are going to do to make the amount of chloride ions that is coming in through this dendritic spine into the neuron greater. Okay, so basically benzodiazepines have a separate binding site on certain GABA-A receptors. Okay, so on the alpha-beta-gamma GABA-A receptors, okay, some alpha-beta-gamma GABA-A receptors will have a benzodiazepine binding site in between the alpha and the gamma subunit. Now, the requirements to have this benzodiazepine binding site, which is often abbreviated, by the way, as BZD for benzodiazepine, okay, binding site, okay, the requirement to have a benzodiazepine binding site is that the gamma subunit absolutely must be gamma-2. No ifs, no buts. If you want a benzodiazepine binding site, that gamma subunit has to be gamma-2. Okay. Now, on our main type of alpha-beta-gamma GABA-A receptor, the alpha-1, beta-2, gamma-2 GABA-A receptor, indeed the gamma subunit is gamma-2, so that's certainly true for the most common GABA-A receptor. Okay, now, as far as the alpha subunit goes, you either need to be alpha-1, alpha-2, alpha-3, or alpha-5. Okay, so alpha-4 and alpha-6 do not cut the slack, basically. Okay, they are not good enough. You need an alpha-1, an alpha-2, an alpha-3, or an alpha-5 if you're going to have a benzodiazepine binding site. So if you have a GABA-A receptor of the alpha-beta-gamma type that has one of these alpha subunits paired with one of these gamma subunits, well, this gamma subunit here, gamma-2 subunit, then you'll have a benzodiazepine binding site. What can happen is the benzodiazepine molecules can bind in this benzodiazepine binding site. So, for instance, diazepam, clonazepam, chlordiazepoxide, okay, all of our benzodiazepines, they can bind here. Okay, so I'll put this in. The benzodiazepine will bind in the benzodiazepine binding site, And then the change that happens is once the benzodiazepine is bound in the benzodiazepine binding site, the GABA-A receptor will undergo a conformational change, and it does not, repeat, it does not cause it to open. Instead, it causes changes in the GABA binding sites here and here. Okay, and it makes these GABA binding sites much higher affinity for GABA. Okay, so the affinity of these GABA binding sites is going to go up, up, up. Okay, now what does that mean? Well, the affinity means how much GABA wants to bind here. Okay, so you know, when you've got a GABA molecule floating around, why does it bind there? Well, it's because it's going to get stuck on there, effectively. It's going to form interactions in that GABA binding site, which are going to attach it there. Okay, now how strong those interactions are will determine how long the GABA stays there before it falls off. Okay, if you have a higher affinity, it means that the interaction between the GABA molecule and the GABA binding site will be much, much stronger. So firstly, the GABA molecules will bind there much quicker, and they'll stay on there for much longer. Okay, so that's what the benzodiazepines, by binding to the benzodiazepine binding site, do. They increase the affinity of GABA for the GABA binding site. So they change the conformation of the GABA binding site so that GABA will bind there with a higher affinity. Okay, now what does this mean? Well, it means that now when, if we go back to our synapse here, if this synapse has now been doused in benzodiazepines, let's say the benzodiazepines are bound to all of the GABA-A receptors here, okay, now the GABA-A receptors, when GABA gets released, you're going to get GABA binding to much more of the GABA-A receptors. Previously, you know, when the GABA was released, you know, some of the GABA-A receptors will end up getting GABA bound to them, but not all of them are necessarily going to get GABA bound to them. Okay, now when you get the GABA released, because of the much higher affinity, a much higher fraction of them are going to get GABA bound to them, okay, which means that a much bigger number of them are going to be opening, and therefore you're going to get more chloride anions coming into the cell. So basically what you do is you increase this inward current of chloride anions, because now you're going to have a greater number of the GABA-A receptors here actually getting GABA bound to them, because they've got a higher affinity for binding the GABA, so they're more likely to bind to the GABA. Okay, and that now means that you're going to tip the balance. The inhibitory currents are going to be much, much bigger. Okay, and that means it's going to be much, much more difficult to activate all the neurons of the brain which have been exposed to the benzodiazepine. Okay, so all the neurons of the brain start firing less often. Okay, now the reason that that is believed to help anxiety is, well, anxiety was caused by overactivity in the amygdala. If we are giving a drug which reduces the activity of neurons all over the brain, then it's going to decrease the activity of the neurons in the amygdala as well. Okay, and therefore it's going to bring the activity of the amygdala down. Okay, so it's a pretty brutal approach to actually doing it, and these drugs they're not the first line treatment for anxiety anymore, they are the second line treatment, but they do work incredibly fast, which is the advantage that they have over the antidepressants. Okay, the disadvantages of them are that firstly, it's easier to overdose on them, okay, and overdoses can be very, very serious. Okay, the other disadvantage of them is that they make you very drowsy and tired. Okay, and indeed they are used as a hypnotic, a sleep-inducing drug. Okay, so their sedative actions are often viewed as a side effect when you want them for the treatment of anxiety rather than the treatment of insomnia. Okay, right. So that now concludes our discussion of anxiety and the anxiolytic drugs.